Imagine a patient comes in, head and neck squamous cell carcinoma.
Okay.
You do the scans, you check the lymph nodes, and, well, physically, they look intact.
Right. No massive tumor you can see.
Exactly. The margins look clean. So you might think, okay, we caught it early, before it spread. But what if that lymph node, even though it looks fine on the outside, has already been completely compromised on a cellular level?
That is the terrifying reality of what we're now calling metastatic tolerance.
Metastatic tolerance.
Yeah. We used to think of the lymph node as, a passive trap.
Like a drain filter.
A drain filter, exactly. It just catches cancer cells. But this new research suggests the tumor is actively terraforming the lymph node.
Terraforming. I like that word. It's not passive at all. It's an engineering project.
It is exactly that. And that's what we're doing a deep dive on today. A paper in Cancer Cell from 2026 by Haist and colleagues.
And the title is a bit of a mouthful.
It is. Its lymph node colonization induces tissue remodeling via immunosuppressive fibroblast-myeloid cell niches, supporting metastatic tolerance.
Okay, let's break that down. Essentially, we're asking, how does a tumor in, say, the throat convince a lymph node in the neck to just turn off its alarm system?
And not just turn it off, but roll out the red carpet.
Wow.
And the answer isn't just about the cancer cells. It's about the whole neighborhood they build. This study is massive, by the way.
How big are we talking?
78 patients, paired primary tumors and lymph nodes, mouse models for validation, and over 1.5 million cells analyzed.
Okay, so before we get into the how, we have to talk about how they even saw this. Because you can't see this kind of architectural change with a standard biopsy, right? Or bulk sequencing.
No, absolutely not. And this is a really crucial point. Standard bulk sequencing is like putting a skyscraper in a blender and analyzing the dust.
You know you have steel and glass, but you don't know the shape of the building.
Exactly. You lose all the spatial context.
Yeah.
And in biology, location is everything.
Function follows form.
So these researchers used a technology called CODEX.
Co-detection by indexing.
Right. Think of it as high-definition cellular mapping. It keeps the tissue intact and lets them see who is sitting next to whom.
So you're not just counting T cells.
No.
You're seeing if that T cell is shaking hands with a dendritic cell or if it's getting cornered by a fibroblast.
That's the perfect analogy. And when they applied this, they found these recurring patterns, these cellular neighborhoods or CNs.
10 of them, I think.
10 distinct types. But there's one in particular that is really the villain of this story.
Okay, let's introduce the antagonist, CN1.
CN1, the myeloid/cancer-associated fibroblast niche. It's basically a toxic clique of cells.
So who's in this clique? Let's get into the specifics, the cell types, and the markers.
Okay, so you have two main partners in crime. First, you have these immunosuppressive myeloid cells.
Macrophages.
Specifically macrophages. But these aren't your typical eat the bad guys macrophages. They're expressing very high levels of PD-L1 and IL-10.
Okay, so PD-L1 is the don't eat me signal. It's a brake on the immune system.
Right.
And IL-10 is an anti-inflammatory cytokine. So these macrophages are basically acting as pacifiers.
They are. And their partner in this clique is the cancer-associated fibroblast, or CAF.
The builders.
The builders. But instead of building healthy tissue, they're expressing periostin and, again, IL-10. They're building a fortress of suppression.
So you have the pacifiers and the builders locking arms. Why is that combination so dangerous?
It's all about where it forms. The spatial mapping showed that this CN1 niche doesn't just hang out on the sidelines.
It moves in.
It physically invades the lymph node follicles and the parafollicular T cell zones.
And the T cell zone is supposed to be the war room. That's where the immune response gets planned and launched.
Exactly. Imagine a SWAT team is trying to brief for a mission, and a construction crew just starts pouring concrete and pumping sleeping gas into the room.
That's a pretty clear image.
That's what CN1 does. By just physically being there, it creates a zone where T cells are immediately shut down.
And do we see that in the data? Are the T cells actually dysfunctional?
Oh, yeah. The study shows the T cells in these metastatic nodes are exhausted.
Meaning they have high PD-1 and LG3.
High PD-1, high LG3. These are the receptors that tell the T cell to stop attacking. At the same time, the regulatory T cells, the TREGs, are hyperactivated.
So the attack dogs are muzzled.
Right.
And the peacekeepers, the TREGs, are shouting, "Everyone calm down."
That is the perfect summary of what's happening.
Okay, we're about 5 minutes in, so let's just pause and recap the key pillars we've set up so far.
Sure.
Point 1, we're using this technology, CODEX, that lets us see the cell neighborhoods.
Right. Spatial context is key.
Point 2, The villain is this neighborhood called CN1, which is a toxic team-up of PD-L1 positive macrophages and these periostin positive fibroblasts.
The pacifiers and the builders.
And point 3, their crime is spatial invasion. They move into the T-cell zones and pump out suppressive signals like IL-10, turning everything off. So here's a question for you listening. If the architecture itself is the problem, if the building is corrupt, can a drug that just targets a single protein really fix it? Or do we need to, demolish the whole renovation? Keep that in mind, because apparently it gets worse.
It really does.
Yeah.
Because everything we just described, you'd assume that only happens once the lymph node is full of cancer.
Yeah, that makes sense. The invasion happens, the city falls.
But the research team looked at lymph nodes that were visually completely tumor-free.
Okay.
They look at clean parts of a metastatic node and even at totally benign nodes from patients who had tumors somewhere else.
And what did they find?
They found CN1. The toxic niche was already there.
Wait. So the construction crew is building the fortress for the enemy before the enemy has even arrived?
Correct. And that is the very definition of metastatic tolerance. The study showed a clear gradient.
A gradient.
The T-cell dysfunction and the presence of that niche were highest in the tumor-filled node, of course. But they were significantly elevated in the adjacent tumor-free nodes when compared to a healthy person.
That's good. That is spooky action at a distance. How does a tumor on the tongue control the architecture of a lymph node centimeters away?
It has to be sending a message.
A signal.
It's chemical warfare. And they identified a specific loop involving 2 key molecules, TGF-beta and CXCL10.
Okay, TGF-beta first. Transforming growth factor beta. Normally that's for wound healing, right?
Yes, exactly. It tells cells to calm down, repair tissue, stop inflammation. The tumor completely hijacks this.
How?
The primary tumor and the fibroblasts start pumping out TGF-beta. It travels to the lymph nodes and forces the macrophages to switch teams.
They go from M1, the pro-inflammatory killers.
M2, like the anti-inflammatory healers. So the tumor is basically screaming, I'm a wound, heal me, and the macrophages obey.
And that suppresses the immune system?
It's a huge part of the suppression. And TGF-beta also activates those ICOs positive tregs we mentioned earlier.
So it's a double whammy.
A double whammy of suppression.
Okay, that explains the stand-down order. But what about CXCL10? I thought CXCL10 was one of the good guys. Doesn't it recruit T cells to the fight?
And that is the paradox that really blew my mind in this paper.
Yeah.
You're right. Classically, CXCL10 is a siren call for effector T cells.
So high CXCL10 should mean a better prognosis. It means the cavalry is on its way.
Usually, yes. But in this specific context, high CXCL10 correlated with worse outcomes.
Why? What's the twist?
The twist is the context. It's the other signals that are present. They found very high interferon gamma signaling in these nodes.
But isn't interferon gamma also a good thing, an activation signal?
It can be. But when you have this chronic low-level interferon gamma mixed with that TGF-beta environment we just talked about,
something changes.
It triggers the cells in that CN1 niche to express PD-L1 and another molecule called ID-L1.
Okay, let me try to walk through this. CXCL10 is the siren it calls the T cells in. The T cells rush to the lymph node, but as soon as they get there, this interferon TGF-beta mix means the niche just slams them with PD-L1.
It's a trap.
It's a wood chipper.
It's a wood chipper. It lures them in just to neutralize them. That's why high CXCL10 is bad here. It's just feeding more troops into the grinder.
That is incredibly sophisticated and, frankly, terrifying. But this is still a correlation. Did they prove this mechanism actually causes the metastasis?
They did, and this is where the mouse models were so critical. They used the B16F melanoma model, which allowed them to watch this happen over time, and they focused on the gatekeepers.
The gatekeepers. These are the CD169 positive subcapsular macrophages.
That's them. So picture the lymph node. It has an outer rim, the subcapsular sinus.
Okay.
Lining that rim are these CD169 positive macrophages. They are the sentries. Their job is to grab bits of tumor antigen from the fluid and hand them off to dendritic cells to sound the alarm.
So they're the first line of detection.
They're the tripwire. And in the metastatic nodes?
Let me guess. They're gone.
They were deleted.
Deleted, like just killed off.
Physically removed from the niche. The study showed a direct inverse relationship. As that toxic CN1 niche expands, the CD169 positive macrophages disappear.
And the traitors, the CN1 cells, take their place on the wall.
And the functional consequence is that antigen presentation just stops.
Yeah.
The dendritic cells don't get the intel, so they can't prime the T cells. The immune system is effectively blinded.
Wow. Okay, we are about at the 10-minute mark. Let's do another quick recap because that was some heavy molecular detail.
Sounds good. Let's lock it in.
Mechanism 1, the long-range signal. The tumor uses TGF-beta to remodel distant nodes,
Forcing macrophages into that suppressive state.
Mechanism 2, the trap. CXCL10 lures T cells in, but this chronic interferon environment means they're immediately neutralized by PD-L1 and IDO1.
Right, the wood chipper.
In Mechanism 3, the blindfold, the gatekeeper CD169 positive macrophages are physically deleted, cutting off the alarm system.
They're replaced by the toxic niche.
So, a question for you listening. If the gatekeepers are gone and the trap is set before the cancer cells even arrive, does that mean metastasis is inevitable? Is the door just wide open?
It certainly implies the door is unlocked and the security cameras are off. But the silver lining here is predictability.
Meaning, if this remodeling happens early, maybe we can spot it early.
Exactly.
Which brings us to the clinical application. Can we look at the original primary tumor and predict if this renovation project has happened in the lymph nodes?
The authors say yes. They found a specific transcriptional signature in the primary tumors of patients who later developed metastasis.
What are we looking for in that signature?
You're looking for high expression of those key signaling molecules we talked about, CXCL9, 10, and 11. And another one, VEGF-C.
VEGF-C, vascular endothelial growth factor C. That's the one that builds lymphatic vessels, the highways for cancer to travel on?
It builds the highways. So you have the highway builder, VEGF-C, and the siren call, CXCL10.
A dangerous combination.
But they also found a spatial signature, something they call the two-chain motif.
Explain that visual. What is a two-chain?
It refers to a specific linking of those cellular neighborhoods we mentioned. In the high-risk tumors, they saw the tumor boundary, which is that toxic CN1 niche,
Right.
physically touching or linking with the B cell zones, which is neighborhood CN4.
And why does it matter if the tumor boundary is touching the B cells?
Because it suggests the tumor is actively corrupting the local immune hub. It's a sign of a very communicative, very manipulative tumor that's already dismantling the local defenses.
So if a pathologist sees this two-chain motif and high CXCL10 in the primary tumor biopsy,
They can say with some confidence, this patient's lymph nodes are likely compromised, even if they look clean on a PET scan.
and that changes the whole treatment plan.
Yeah.
This is the big so what then. How do we treat this? Because if the lymph nodes are already poisoned soil, just cutting out the tumor feels insufficient.
That's the author's core conclusion. They argue really strongly for neoadjuvant therapy.
Neoadjuvant meaning you give the drugs before you do the surgery.
Right. You have to try and normalize the tissue environment before you go in with a scalpel. And they suggest a specific combination based on these mechanisms.
Okay, let's build the combo. We know the T cells are exhausted by the PD-1, PD-L1 pathway.
So you use an anti-PD-L1 drug, standard immunotherapy.
Right. But we know that Oakbiders often fails in these patients on its own.
And now we know why. It's because of the fibroblasts. It's that whole CN1 niche protecting the tumor.
So you have to break down the fortress,
You have to break the fortress. So the researchers propose adding a TGF-beta blocker.
which would stop the signal that turns the macrophages bad and activates the fibroblasts in the first place.
Exactly. In their mouse models, this was really effective. Blocking TGF-beta prevented the toxic niche from forming. It allowed the gatekeeper macrophages to survive.
And once the fortress is down,
The anti-PD-L1 can finally do its job.
you fix the soil so the seed of the immune response can actually grow.
Precisely. You turn off the trap, so the T cells that get recruited by CXCL10 can actually kill the cancer instead of being killed themselves.
That is a massive shift in thinking. It's not just kill the cancer cell. It's restore the entire ecosystem.
It really pushes oncology towards systems biology. We're not just hunting a single mutated cell anymore. We're trying to repair a corrupted communication network.
Let's summarize the entire cascade one last time, just step by step, to make sure it sticks with everyone.
Okay, step 1. The primary tumor secretes TGF-beta and VEGF-C.
Step 2. Those signals travel to the lymph node and build the CN1 toxic niche.
Made of the PD-L1 positive macrophages and the CSS.
Step 3. The gatekeepers, the CD169 positive macrophages, are deleted.
The alarm is cut.
Step 4. The tumor then secretes CXCL10, which lures the T cells into the node.
The trap is set.
Step 5. The T cells arrive, but they're immediately hit with PD-L1 and IDO1 from the niche, rendering them useless,
They're neutralized.
which leads to Step 6. The lymph node is now a safe haven, prepped and ready for the cancer cells to metastasize into.
It is a brilliantly evil strategy by the cancer. But, knowing the strategy is the first step to beating it.
Absolutely.
Yeah.
And being able to predict this from the primary tumor signature is a potential game changer.
For personalized medicine, yes.
So we'll leave you with this final thought. We always talk about clean margins in surgery. Did we get all the cancer? But this research suggests that clean is just a visual illusion.
It suggests the ghost of the tumor inhabits the tissue long before the tumor itself even arrives.
The lymph node isn't just a passive drain. It's a battlefield where the architecture itself, the very beams and walls of the building, can be weaponized against us.
And maybe the future of curing metastasis isn't just about better poisons, but about becoming better architects.
Thanks for listening to this deep dive into the remodeling of the immune system. We'll see you next time.